So hopefully not too many of you noticed how many unread emails I have. When you're looking at the screen there earlier, it's a little on the embarrassing side, to say the least. I try and keep the last month's worth of emails on my first page, but it doesn't always work. So second thing, clickers. I finally figured out the software. It had to do with incompatibility between the iClicker software and the OS on my computer. So many of you, not all of you, should have scores now in D2L. So I see some nodding heads. Good. If you think you have registered and you're not seeing scores in D2L, send me an email with your name, your clicker number, and your username. But again, check there first, re-register first. I'm getting all of the scores. I've got lots of clickers that nobody has actually registered to yet, so I've got lots and lots of those registered. It's just, you know, I don't have a connection to your name yet. So as long as that happens, actually even by the end of the course, it's fine. But best if we can get that done by the first midterm. Any questions about that sort of, you know, right now? I've also figured out that I need to go two-fisted here with two different remotes. Don't ask me why. It's also fun with technology. So what we talked about last time was chemistry. Again, I'll usually try and do a quick review, first slide or so of the following lecture on what we did last time. We talked a little bit about all the atoms in biology. Fortunately, there are not that many of them. Hydrogen, nitrogen, carbon, oxygen, phosphorus, and then, you know, a little bit of some of the other ones as well, sulfur, etc. What we are really going to care about mostly for this course is going to be the weak interactions, and the weak interactions are important for two reasons. One, that they are reversible, so you can have dynamics, and two, that you can have lots and lots and lots of them, and they can vary a whole bunch. So you can really be quite flexible, and that's what biology really needs is flexibility. And it's lots and lots of them coming together. So van der Waals interactions, hydrogen bonding interactions, ionic interactions, and then hydrophobic interactions. And quotes for hydrophobic interactions, because it's all about water liking to interact with water. And if water is interacting with water by forming hydrogen bonds, then it's going to force anything that's not interacting with water together. And we're going to spend most of today talking about what things those happen to be, and it's mostly the side chains of amino acids, which are going to be in proteins. Now, this only works in terms of having lots of these small, weak interactions if you have large molecules that are going to be able to have lots of these interactions. So in order to do that, you also are going to have some strong interactions, and that's almost always going to be covalent bonds as far as we're concerned for this class. So how do you make covalent bonds? It's actually really kind of difficult to do that from an energetic point of view, and so you need to have enzymes that will help in terms of getting your reaction to go faster. Doesn't change the actual reaction per se, but it allows it to go faster, and in that process lowers an activation energy barrier so that all of us don't, you know, spontaneously combust. The next thing that biology has figured out how to do, you know, evolutionarily, really fascinating questions about origins of life and where this came from in the first place. We're not going to talk about that here, but the idea of having a high energy bond, and particularly a high energy intermediate. So you go from one molecule, have a favorable reaction, and then have a unfavorable reaction, which is then coupled to that previous reaction. And very often, again, this is going to be some kind of nucleotide triphosphate. We're mostly going to be talking about ATP in this class. And so that process, using the high energy bonds that we have in, again, usually adenosine triphosphate between the three different phosphates. We'll talk more about that when we talk about DNA structure next week. And then the cleavage of those bonds, which then was coupled to the formation of other bonds. And I just wanted to do a quick shout out to the textbook. And yes, it's ridiculously heavy and expensive, etc. But one very useful place to go and look for review items is in this little blue section right here, all these different panels, pages 90 to 102 in the 6th edition of the textbook. So nice place to go, again, for pretty concise review of everything that we talked about in terms of chemistry. So more chemistry questions. Going once, going twice, gone. When he says, if you have any questions, what's he going to do? Ask you a clicker question. So almost all polymerization reactions are in biology. Which of the following reactions? Acid-base catalysis, condensation, hydrolysis, dehydrogenation, or reduction? And again, spend a minute, figure out what your answer is. If all of you have it correct, then we're good. If not, then you'll have a chance to chat with your neighbors about it. Okay, we have not reached a consensus, so tell your neighbors what you selected and why. Ready to go again? Yes? No? Go. Sure.
Nobody around me knows, but nobody else is.
Mix and match. No. Well, between you can all. I guess how many you got? 5 around you, so each you can choose one. Okay, I don't see any more people voting, so. Okay, let's see what people think. B, tell those people around you that B's right. So the key here is polymerization reaction. So any kind of polymerization reaction, what are you going to be doing? You're going to be taking water out. And water is always going to be involved in these reactions. So hydrolysis is going to be the opposite of condensation, exactly. So now move forward with this, point with this in theory. So talk about proteins, and again, this does connect very closely to the stuff that we were just talking about in terms of hydrophobic, hydrophilic, etc. So proteins are basically polymers of amino acids. Let's talk a little bit about the different amino acids. No, I don't expect you to memorize all their structures. No, I don't expect you to memorize all of the single letter codes for them. I will try and write them out if we ever use them on exams. But there are 4 amino acids which are particularly important that we'll spend some more time talking about. Get to those in just a second. Amino acids fit together with peptide bonds. That's how the polymerization works. Again, these are covalent bonds. They're all made by the ribosome. Once you have hooked up these individual amino acids together, we call that the primary structure. Also has a molecular biological polarity to it. It's got a start and a stop. That secondary structure forms through hydrogen bonds and hydrogen bonds between the what are called side chains of the amino acids. And those are the things that are sticking out, or between the backbones of the amino acids. So, sorry, back up a little bit. Secondary structures are all backbone hydrogen bond interactions. So the tertiary structures, which is protein folding, which come from the side chain interactions. So secondary structures you can form with any particular amino acid. And there are 20 what we call canonical amino acids. There are a few different ones that we'll probably talk about when we talk about translation a little bit later on. Once you have all of this protein folding, all the secondary structures are going to fit together to give you a final stable structure, and structures give us function. Good. Well, by the end of the term, you'll all have it down. A couple of things about these structures. Most proteins that we're going to talk about have actually multiple structures. And if you've got multiple structures, that means they're going to have multiple functions. Good. And those separate pieces, separate structures of the proteins are called domains. And it turns out that lots of proteins have different domains, and each of those domains usually is going to have one particular kind of activity, and it's the combination of those domains which will give you potentially lots of activities. And a really nice example is how do you get coupled reactions, because you got multiple domains of a protein that are involved in coupling reactions. Another thing that many proteins have is called multimers. This seems to be a little bit of a confusing topic. When I say a multimer, anytime I talk about a single protein or a single polypeptide chain, I will use those interchangeably. Probably not really good, but I end up doing that. They will have one N terminus and one C terminus, so one beginning and one end. Anytime you've got more than one N terminus and more than one C terminus, that's going to be a multimer. And it turns out that many, many, many proteins are multimers, particularly the very important proteins that we're going to be talking about in this course, the polymerases, some of the other proteins as well. But very often, these individual polypeptides come together to form a structure which will then have a particular function. I kind of skipped over motifs here. Should mention this as well. This seems to be something that I do a really bad job of explaining, I guess, most of the time, because people don't seem to follow it that well. Motif is just a primary structure, so a sequence of amino acids that gives you some indication it's going to have a particular function. But motifs in and of themselves do not have structure. So a motif is going to be part of a larger structure or domain or something like that. And we'll talk a lot about DNA binding motifs. We'll talk about dimerization motifs, but they're motifs. They're protein primary sequence. They're not an actual domain. Talk a little bit about protein families and maybe have a chance to talk about modeling right at the end. So first, amino acids. Let me move this out of the way for the time being now. Amino acids, just a single amino acid also has a amino terminus and a carboxy terminus, a start and a stop. This is your backbone. There's always going to be one carbon between the amino group and the carboxy group. Hanging off of that carbon is the variable side chain. This is what gives you the different amino acids. So this particular one, methionine, this has got a different side chain, aspartic acid, leucine, tyrosine, etc. The peptide bond forms between the carboxy group and the amino group. What happens in this reaction? It's a, what kind of reaction? Condensation reaction, exactly, thank you. And you form these together, is this going to be energetically favorable, energetically non-favorable? Unfavorable, exactly. So be coupled to something else. We talk about translation. We'll talk about that later. So this is probably the best way to look at peptide structures. But since everything's going to be the same here, this amino group is going to be the same as this carboxyl group, or I should say this peptide bond, etc. What we really care about are what's going on with these individual side chains. And so people will often use these schematically with these blobs sticking out along the backbone, or very often just a sequence, as we have down here at the bottom, met, asp, lu, tire, for instance. That will be the process involved there. Let's see, the pointer here seems to have died. Another way of looking at, so hang on, we went too far. I have to call these different flavors of amino acids. Looks like the pointer has died here. Sorry about that. See, always when I try and do my different technology, it's going to be a little bit different. Different flavors of amino acids. So again, we talked about hydrophobic, hydrophilic, and basically what each of these amino acids are actually doing. There's really two big groups, not surprisingly, those that like to interact with water and those that don't like to interact with water. And again, this is our, switching gears, these are our chemical polarities. So a polar amino acid is one that likes to interact with water. A nonpolar amino acid does not like to interact with water. I like to call these your hydrophilic and hydrophobic amino acids. Why they call them polar and nonpolar, again, I try and keep polarity for our molecular biology polarity here. The ones that we're going to be most interested in are going to be these, those that acid, acid, base, base. And another way of looking at this, which is actually far more information than you ever need to know about any of the individual amino acids. Unplug this, see if that will help. There we go. So these are, again, all of the amino acids, all 20 of the canonical ones, I should say, and basically all the things that they do. Again, I don't expect you to memorize this at all. One thing that I find is very useful are the single letter codes for each of these amino acids. Again, I don't expect that you will remember them, but if you're ever going to continue with this, it'd be a good thing to learn. They're pretty straightforward. V is valine, I is isoleucine, T is threonine, but you've also got some funky ones, W for tryptophan, F for phenylalanine, Y for tyrosine, etc. Again, the ones that we really care about are K, D and E, arginine and lysine, aspartic acid, and glutamic acid. And why is that? That's because these amino acids can stably ionize, and in fact they do at neutral pH. So most of the time, and this is going to depend exactly where this particular amino acid is in your protein, all of the acidic amino acids will have given up a hydrogen, which is what we talked about last time as being what defines your acid. And so they will have a stable negative charge. And this is now on the side chain. All got the regular peptide backbone down here. Side chain is stably negatively charged at neutral pH. On the other hand, the basic amino acids, those are the ones that are going to pick up hydrogen. Here now we have a stably positively charged. So lysine and arginine, also known as K and R, are going to be your positively charged. Glutamate and aspartate, or glutamic acid and aspartic acid, are going to be those which have stable negative charges. And again, these are the ones that I will expect you to know. Because, as we'll see again later on, very important in terms of different interactions, both in terms of protein folding, but also in terms of interactions with nucleic acids. So, already mentioned, hopefully a kind of, one of my students before sent me this little GIF of a horse and somebody with a baseball bat, so beating the dead horse. So hopefully some of that is not too extreme here. Primary structure is just the sequence of your individual amino acids. How do you tell the difference between those amino acids? It's all going to be the side chains, shown here as these big green blobs, and then the peptide backbone in between. Very rarely are you going to have a protein that actually has something all nicely lined up like this, but this is what we can figure out from the sequence, and sequences is easy, structures are hard, so this is what we start with. These primary structures then form a number of different secondary structures, and we'll look at those, the major ones, alpha helices and beta-pleated sheets, in just a second. These are formed by hydrogen bonds, these little dashed lines here, the red dashed lines, between backbone oxygens and nitrogens. Again, sharing this hydrogen atom right here. Secondary structures in and of themselves are usually not very stable. They have to then interact with other secondary structures, giving you a tertiary structure. And so this tertiary structure, this is what's a nice three-dimensional, usually pretty stable. We'll talk about unstable proteins again later on. It's always exceptions. This is biology. But that tertiary structure is what's going to give you your stable structure, your stable activity. In some cases, and this is also the multimer effect, you can have multiple polypeptides. So here we've got one end, the other end must be hidden in here somewhere, that then comes together. In this case, we've got a tetramer. You've got 4 polypeptides coming together. Again, this is not in all cases. All proteins are going to have some kind of tertiary structure at some point. A few of them will have these quaternary structures. How does this actually happen? It's really about interactions between the side chains of the amino acids, and usually what happens, again, it's biology, there are always exceptions, is those that are the hydrophobic amino acids end up in the middle of your protein, and the hydrophilic amino acids end up on the outside in your tertiary structure. And this is also called the folding process, where you go from a linear stretch of amino acids to this three-dimensional structure down here at the bottom. We can look at protein structures in lots of different ways. These are sort of the 4 classic ways of looking at them. The probably simplest way of looking at an amino acid, so sequence and then in a tertiary structure, three-dimensional structure, you can just look at the backbone, and that's shown here. All of these are color-coded from red at one end to blue at the other. And I don't have actually no idea which one is N-terminus and C-terminus. I think this is one N, that one C. Doesn't really matter. And what we're mostly going to look at in this class is this sort of cartoon version where you have secondary structures like this alpha helix and these 2 beta strands coming together. You could also, and this is people call ball and stick diagrams or more stick diagrams, have each of those separate amino acid side chains sticking out. I find this is very confusing to look at, but it does tell you some things like this here, for instance, aromatic amino acid here. What other proteins see and what other nucleotides see is these are the van der Waals radii, and remember, very fine defined distance between the nucleus and the electrons of the atom, depending on where you're going to have van der Waals interactions. So if you think about how different proteins are interacting with each other, how different proteins are interacting with nucleic acids, or even individual amino acids interacting with each other, this version down here is probably the best one to use. However, what kind of secondary structures are there in here? You can't tell from this. And so how each of the pieces are organized, it's usually easier to look at like this. So we're usually going to be looking at protein structures, and almost all of the figures in the textbook are going to be looking at this, where you've got your alpha helices, your beta sheets, and then these extra loops between them out here. So let's look at a couple of the classic secondary structures. The alpha helix, originally proposed by Linus Pauling from where? Portland, Oregon. Yay! One of the very few people to get 2 Nobel Prizes. And in fact, in 2 completely different fields, I think he was the only one to end up getting 2 Nobel Prizes, in chemistry and the Peace Prize. And if you go down to Corvallis at any point, any of you have been there, the Linus Pauling Science Center, really nice building, way nicer than the science buildings on this campus so far. We're working on renovating Science 1, different story. But he was the one who came up with the concept of the hydrogen bond. And then in looking at hydrogen bonds and what he knew about the individual amino acids, he came up with both structures for, or I say proposals, really. These were theories about how alpha helices should fit together and how beta sheets should fit together. Turned out to be almost exactly correct. And he found some proteins that were mostly alpha helix and mostly beta sheet, and was able to show that they certainly had these kinds of helical structures. But it was really the breakthrough in thinking about hydrogen bonds, how big these hydrogen bonds were, and if you had hydrogen bonds, again, between backbone carbons, sorry, I said nitrogens, oxygens, and hydrogen. Again, this hydrogen bond here, the dashed line, you could form these alpha helices. And one of the really nice things about the alpha helix is where are all of these R's or side chains? They're sticking out. Where are you getting specificity? Interactions with other proteins, other parts of the same protein, nucleic acids, it's going to be on those side chains that are sticking out. So the alpha helix is great because it's sticking side chains out on either side, and it doesn't matter what those side chains are, you can still form an alpha helix. And somebody over there is probably going to hold their hand and say, Dr. Stedman, what about prolines? So prolines are weird. They actually don't have these side chains. The side chain actually comes back and loops back to the carbon in the middle. So you can't make alpha helices with prolines. But again, it's biology. There are always going to be exceptions. But those 19 other amino acids, you can use them to make these alpha helices. We'll talk more about this when we talk about the DNA structure later on. The alpha helix is a right-handed helix, or I like to think about it here in this kind of scale as a right-armed helix. So if I reach up, it's going to be going around with my right arm. If I try to do it with the left arm, it's wrong. So when I bring my DNA model, we'll start that next week. You'll see that process. Some of you, though, hands. My hands don't work that way. So if you wrap your fingers around, they should be wrapping up towards your thumb. That will give you your right-handed versus your left-handed helix. The other major secondary structure that, again, Pauling came up with was the idea of the beta sheet. Again, this is all about hydrogen bonds between backbone atoms. So again, these dotted lines here, here's the oxygen, here's the hydrogen. The big difference here is instead of, and I should back up here, in this case, the hydrogen bonds that are forming are all in amino acids that are quite close to each other in primary sequence. In a beta sheet, these hydrogen bond interactions can be a very, very, very long distance away from each other in that primary structure in the primary sequence. They just have to line up with each other. And remember, hydrogen bonds are very directional. So alpha helices, hydrogen bonds close to each other in the primary sequence, beta strands far apart, and they can be either in a anti-parallel, and again, this is our molecular biology polarity here, N-terminus going to C-terminus. So the N-terminus here could be forming hydrogen bonds with the C-terminus over here, or you can have parallel beta sheets where it's always going to be the N-terminus that's interacting. In this case, it makes no difference in terms of what orientation they are, because where those hydrogen bonds stick out is going to be in the same place. Everybody happy with secondary structures? So happy you can answer a clicker question on them? Good. Formation of protein secondary structures. So due to which of the following? Van der Waals interactions between side chains, hydrogen bonds between side chains, Van der Waals interactions between backbone atoms, hydrogen bonds between backbone atoms, or hydrophobic interactions? And hopefully this horse is dead by now. Okay, usually I like to see 80% plus get it right, and we're not quite there yet. So I'm hoping that in discussing with your neighbors or further away, if you don't like the people right around you, jump up and run over to the other side of the room. So yeah, what do you select and why? Let's see if we can get ourselves. Eventually we get 100%. That's another one of my goals for this course. We ready? You found somebody further away to chat with? Okay, let's go. And you can continue to discuss now. You don't have to be quiet while you're voting. Clicking the button doesn't mean make a difference. If you're still not sure. Okay, I actually like these numbers much better. So it is between the backbone atoms, not between the side chains. Side chains are going to be important for tertiary structure, which is what you're just about to talk about. And I've got to come up with some other questions for the some other possible answers for the other things. It turns out that all of the other ones between the side chains are what is going to be important for tertiary structure and not for secondary structure by themselves. So yes, it is D. No, not to. Okay, so got our secondary structures again, hydrogen bonds between backbone atoms. Now we need to get to that tertiary structure, because most secondary structures are not going to be stable by themselves. So you have to get them to come together. This, again, all of those different kinds of interactions that we can now have between the different side chains. You can have ionic interactions between those acidic amino acids, remember, glutamate and aspartate, and the basic amino acids, lysines and arginines. So it's one of the reasons that I keep harping on about these things. You can also have hydrogen bonds that now form between side chains, not between backbone interactions. And here is actually a backbone one. Sometimes you have them, but for the vast majority of the time, it's going to be between the actual side chains. And then polar, nonpolar, green versus blue. So green is going to get packed in the middle, or literally squished into the middle, because the water is going to be interacting with itself and with all of those side chains that are on the outside. And then there's one extra piece here that they don't talk about in this particular section of the chapter, but and for some reason I couldn't find a nice version, so it's really pixelated here. Cysteines. Cysteines are very different. Again, I didn't talk about them per se because we're not going to talk too much about them in this class. Cysteines have a sulfur in them. They're not the only amino acid that has sulfur. Methionine also has sulfur in them. But most importantly, they have a sulfhydryl, or an SH group. And what that means is you can often have these SH groups interact with each other and form what's called a disulfide bond. And what that does is now a covalent interaction that you have between two side chains, often in very different parts of your protein. So this really constrains where that particular part of your protein can actually be. And it turns out that most inside of cells are actually in reducing environments, and in a reducing environment that's actually going to be an SH. So they're not going to be forming disulfide bonds, but as soon as something gets outside of the intracellular milieu, it's going to be an oxidizing environment, and then you'll have these disulfide bonds that form with each other. So they're almost all secreted proteins, and the kind of protein that's going to be on the outside of a cell, very often these are going to have disulfide bonds that are associated with them. So side chain ionic interactions, side chain hydrogen bonds, side chain van der Waals interactions. That was what was supposed to be shown down here at the bottom that I forgot to mention. And then hydrophobic interactions, sort of squishing things together. One thing to mention, again, I talked about helices and we talked about the alpha helix. We've got, again, I like the right-armed versus the left-armed helix. This will be important also when we talk about the nucleic acids later on. How do you make a helix? We already talked about the helix being really neat because in the case of the alpha helix, you're sticking the side groups out on the outside. So that is a really nice way to expose all of these things. Helices are also really nice because they have symmetry to them. Turns out symmetry is really nice for packing things together. How do you make a helix? Basically, you can make a helix if you've got two different interaction sites on, it's actually any molecule. And we'll see this when we talk about the DNA helix later on. Basically, a front end and a back end in terms of these interacting points. And what did we just talk about, front ends and back ends for amino acids? Amino groups and carboxy groups. So they're going to be interacting with each other. They can interact in this helical form, or, and we see if you're talking now about side chains that are interacting with each other, and in fact, in many cases, we'll talk about, see this in just a second, we've got multimeric proteins, got two interaction domains. They can interact in order to form some kind of ring structure. And rings turn out to be also be very, very important in molecular biology, particularly in replication processes. We'll also talk a little bit about dimerization interactions, and so this is where you have the same structure on two individual polypeptides, or actually for that matter, two molecules. Those will like to fit together, but they're going to be the same interactions with each other, so you're not going to form helices or rings if they're just one kind of interaction between each other. Why do we care so much about these things? Well, one of the classic examples of these kinds of interactions, we've got now a combination of a dimer and a helix. So these are what are called coiled coils. The coil itself is the helix, and then the helices are coiled around each other. We'll talk mostly about these in the context of something called a leucine zipper. Why is it called a leucine zipper? That's because every 7 amino acids, you have a leucine or some other large hydrophobic side chain. That large hydrophobic side chain is going to want to interact with what? Other large hydrophobic side chains. And again, if you got an alpha helix, alpha helices, which I forgot to mention before, every 3.5 approximately amino acids, it goes round. And so if you go 7 amino acids, you're going to end up on the same side of an alpha helix. On the same side of an alpha helix, you've got 2 leucines that are sticking out. You've got another alpha helix over here, 2 leucines sticking out. They'll stick into each other. And that's exactly what happens in these leucine zippers. And these are really neat because one of the very few kinds of amino acid sequences that you can look at, just literally at the primary sequence, you've got every 7 amino acids, you've got a leucine or some other large hydrophobic. You go, I wonder what this might be doing. And quite likely it's going to be interacting with either itself or some other alpha helix that also has every 7 amino acids a large hydrophobic. And we'll talk about these in terms of DNA binding proteins a lot later on. And this is very common in terms of a dimerization. Now, this structure in and of itself is actually not stable. So it's a primary sequence that is likely telling you something about function, but it doesn't have a stable structure. What we call it? A motif, exactly. So this is one of the first motifs that we've now talked about. So the coiled coil motif or the leucine zipper motif. These motifs, again, are going to be parts of larger folded pieces, which are called domains. Domains, again, as I mentioned before, this is going to be a separate part of the protein. It's linear in amino acid sequence, but it forms its own structure. And so here's a nice example of a multi-domain protein. How do we know it's multiple domains? Because they give them different colors. But we know that they have separate structures. So for instance, here, this is one domain of your protein. It's got a separate structure. Very often it's going to have a separate function. Here's a different domain down here. Here's a different domain here. Here's a small domain over here. So these domains, when they interact with each other, they're going to give you a slightly different kind of function. And these are some examples down here at the bottom. So each of these colored blocks represents a different domain. These have often very similar kinds of functions. So it turns out chymotrypsin, what does it do? It's a protease. It breaks down proteins. Factor IX, it's a protease. It breaks down proteins. Plasminogen is a protease. It breaks down proteins. But they break down different things. And they break down different things because they've got these different domains that are attached to them. And so these different domains are going to be saying, okay, you're going to interact with this kind of thing that you're going to break down. This one's going to interact with different ones, etc. Sometimes these domains are going to be a regulatory domain. And so it's that mixing and matching of these different kinds of domains and that recombination. We'll talk about the processes of recombination later, but we looked at how you get different genes and different functions. After you have gene duplication, you can have recombination where you can be moving different pieces of proteins around. And what we found is just in looking at now literally thousands of protein structures with multiple domains in them, some domains come up again and again and again. And that's probably because life's been evolving for 4+ billion years, or approximately 4 billion years. And some of these structures actually work. And if it's something that works, you hang on to it. And then you tweak it a little bit. It's way easier to tweak something, particularly if you think about it from a mutagenesis point of view, than come up with a whole brand new thing. So that seems to be why you have all of these domains even though you can have a completely different primary structure, and we'll see this in just a second here, primary structure being this amino acid sequence, still end up with a very similar kind of tertiary structure, and that tertiary structure is probably going to give you a very similar kind of function. So let's look at a couple of examples of these. This one at the top is two different kinds of proteases, elastase and chymotrypsin. They're not very similar to each other at the amino acid sequence level. This case is actually slightly above 30% amino acids, supposed to be shown here by the green segments. So this green segment right here, this green segment right here, very similar structure. In this case, that's actually an identical amino acid at that position. On the other hand, this gray sequence here and this gray sequence here actually are different amino acids. And what this is supposed to be showing is that you end up with these very similar structures. In this case, an almost identical function. Elastase and chymotrypsin actually cut slightly different amino acids and proteases. But few amino acids are the same. Nonetheless, you have a very similar structure. And there's kind of a rule of thumb when you're looking at amino acid sequences, and mentioned bioinformatics last time, the easy way to figure out what your favorite protein does. We look at amino acid sequence similarity. We get an idea if something was homologous. That's great. Got a similar amino acid sequence. What does it mean? How likely is it to have a similar function? Well, it turns out, and again, this is just from looking at lots and lots of different structures, that if you have more than about 30% identical amino acids between two different sequences, they're highly likely to have an almost identical structure. And people have looked at this in a great deal of detail, and it's pretty good. Now, the flip side, which is less than 30% amino acid identity having different structures, that you can't bet on, because that's the example down at the bottom. Here we have two proteins which have almost identical structures, but they're less than 30% amino acid identity between the two of them. And there are actually some really good cases that are not in the textbook, but where you have completely different sequences, maybe 15, 20% amino acid identity, which is basically almost random, and still almost identical structures to each other. Now, one thing that I also wanted to mention here, spent quite a long time talking about paralogs and orthologs. Elastase and chymotrypsin are present in the same organism. So what does that mean if they are sequence related to each other? It can be paralogs or orthologs. Paralogs. What's true about paralogs in terms of their functions? They've got different function. In this case, it's similar, but they're cutting different amino acids. On the other hand, we've got down here two similar sequences in very different organisms. When they say yeast here, it's Saccharomyces and Drosophila. So what are these going to be? Paralogs, likely to have similar or different functions. Paralogs are likely to have, orthologs are likely to have different. Paralogs are likely to have same. So it turns out that in this case, they do have almost exactly the same kind of function. This is a DNA binding protein that's important for development in Drosophila and also in yeast. So orthologs, you're just going to have different functions. Paralogs are likely to have same functions. Okay, just as a reminder, oh, sorry.
I had a question about domains.
Yeah, sure.
Is it accurate to think about domains as being different tertiary structures within the same primary, or is all the domains just one tertiary structure? Does that make sense? So
Not sure I completely understand your question, but I'll try and rephrase it in such a way that hopefully makes more sense. So domains are going to be, one domain is going to be a contiguous stretch of amino acids. And so in this case, for instance, just up here, the teal part. And that in and of itself is going to have a tertiary structure which is stable by itself. That'll very often have a particular function. Now, that function could be interacting with a different protein, could be a substrate, just stabilizing the protein for that matter.
So you can say that domain has a different tertiary structure than that domain.
Yeah. Yeah, so that difference. But what you will also see is in some proteins, and a nice example down here at the bottom, you can have multiple copies of a domain with very, very similar structure. So, and that could be 4 copies of the XYZ domain. This particular case, in the textbook, they spent a lot of time talking about the SH domain, the sarcomology domain. That's partly because one of the authors works on that. So, but yeah, you can have multiple copies of a very similar kinds of structure. Unlikely to have exactly the same sequence, but will have a very, very similar structure. Probably more than 30% identical, which gives you an idea that that's likely to have a very, very similar structure. Yeah, over here.
I'm sorry, I'm just a little bit confused about paralogs and
Orthologs, yeah.
the difference between those two.
Mm-hmm.
Was it that the example on the top was that they were 2 amino acids that have the same structure but have different functionalities between the elastase?
Okay, so there are 2 different things I'm talking about. Sorry, finish your question.
And then I had it backwards on if paralogs or orthologs have the same function or different function.
Okay, well, if you think about it, so I'll try and explain this again. But the idea is that you're going to have orthologs are going to be in the same organism, right? So it's gene duplication going to be in the same organism. Why hang on to 2 things that are doing exactly the same thing in the same organism? So those are likely to be forming differences. The paralogs are going to be between, did I mix them up? Did I mix up ortholog and paralog? I see people shaking their heads. Sorry about that.
We've been whispering about it.
Okay, yeah. Yeah. Which ones you got them forwards and backwards? Yeah. So, but then in different organisms, they're likely to have the same function because that gene duplication has happened, actually hasn't really happened. It's just diverged because they've had this divergent. See, this is why probably lots of people have trouble with this because I keep mixing them up. Yeah.
I feel like it's biologists that happens to all of us, but and that was for paralogs being different organisms.
Yeah, so anything, so if you have gene duplication in the same organism, let's just ignore the ortholog and the paralog. Gene duplication in the same organism and divergence in the same organism, they're likely to have different functions. Whereas if they're going to be similar sequences and similar proteins in different organisms, they're actually quite likely to have the same function. Is that better? Paralogs and orthologs? I'll try and beat this into my head again and get them confused. Yeah, sorry.
On this slide it says protein families, and I was
Yes.
wondering, is that protein that has similar function to another one, or like some percentage of the same type of domain? Or what makes it a protein?
So what makes it a protein family? Why did Stedman put protein families on this slide? To remind himself he was supposed to talk about them, and thank you for reminding me. So these are, again, orthologs and paralogs are all going to be parts of protein families. And how do you know that? It's usually from looking at these sequences. And they'll have similar functions, again, or they'll have almost identical functions. To some extent, that's going to be same organism, different organism. But when you've got all these gene duplications, they're going to have similar sequences, which also means they're going to have similar functions. And so that was why I just wanted to mention the protein family thing, because we talked about protein families before. Thank you for reminding me about that. Okay, quick sort of an aside here. I always need to remind myself of scale. If you think about proteins versus nucleic acids, proteins are really big relative to nucleic acids. But proteins are also really variable in terms of their sizes. You can have some proteins that are really tiny, insulin, issues with insulin supply and et cetera, which we won't talk about. That's way beyond the scope of this course. Hemoglobin, which is a slightly larger molecule. But then immunoglobulins, some metabolic enzymes start to be really, really ginormous. And we'll see for maybe today, maybe on Monday, exactly how big some of these molecules end up being. They're really ginormous, particularly when compared to DNA molecules, RNA molecules. And then, if you think about the fact that we've got trillions of cells in our body, all that stuff's got to get into these cells and still be able to move around and interact with each other. It's kind of mind-blowing that all of this works. So again, we'll talk more about it later, but I just wanted to, it's good for me to think about the fact that proteins are generally pretty darn big, and also relative to a pretty standard lipid bilayer that you can have on the outside of the cell. How do they get so big? Some of them are just literally really, really large numbers of amino acids that are hooked up N-terminus to C-terminus. That's rare that that's the case. Usually, they're going to be multiple polypeptides that are coming together. So again, multiple N and C termini. Dimers are a classic case of this, and we'll talk more about this particular dimer a little bit later on. One green one, one red one. They interact in this particular case through two beta strands that are going to interact again with side chain hydrogen bonds. So I mentioned before, you can have beta strands interacting with each other that are very long distances apart in sequence. Well, in this case, it's an infinite amount of sequence between the two because they're actually in two completely different molecules that they're interacting with each other. You can also have tetrameric proteins. This is 4 polypeptide chains that have come together. This particular one, neuraminidase. How many of you heard about H5N1 or H1N1? What are they? Flu. All gotten our immunizations? Yes, good. I won't force you to hold your hands up, but those are the people you don't want to be sitting next to who haven't gotten immunized. But one of the best functioning anti-influenza drugs actually interacts with this particular protein and interacts with it right here between these individual, and often I'll talk about subunits of some of these proteins. Also, this is where the activity is in many of these proteins. The largest multimer that we'll talk about here, I think actually it's probably the largest multimer that we'll talk about in this class, actually has 12 different subunits, 12 different polypeptides that come together for one particular activity. This is the RNA polymerase, and cartooned up here in the corner, 1, 2, 3, 4, et cetera, et cetera. Every single one of these polypeptides is required for the activity of RNA polymerization. And if we looked at this in the scale slide back here, this would be even bigger than glutamine synthetase. Yet it's still sitting down on DNA and making RNA. So just again, examples of some of these things. And later on in the course, I'll pull up a bunch of structures. You'll see them a little structure dependent on some of these things a little bit later on. Now, structured, right? All proteins have structure. Well, yeah.
Quick question before we move on.
Yeah.
Do they tend to link together primarily through interactions with the backbones or through the side chains?
These are almost always going to be interacting. The question was backbone interactions or side chain interactions. Backbone interactions, it's actually very rare that you have those between different protein subunits. It's going to be much more common that it's side chain interactions. Because that's where you get specificity. If you think about backbone interactions, those aren't specific. Every single amino acid has one. Whereas if you want to have some kind of specificity, it's going to be those different side chains. Yeah?
Are they typically covalent, like disulfides, or can they be weaker?
So there are a few cases, and it's actually almost exceptions, where you have disulfides between these different subunits. Usually there are lots and lots of the non-covalent interactions between them. One big exception is actually antibodies, the immunoglobulins that I showed. But for the most part, it's usually not. And it's also partly because many of these things are acting inside the cell, where cysteines are going to be, it's a reducing environment, so they're not going to be even forming disulfides. Okay, so I'm going to finish up talking about non-structured proteins. One of the, again, big surprises that came along once we did some of the science is that quite a lot of proteins, and actually quite a lot of parts of proteins, don't have a stable structure. And Monday we'll talk about how we figured out how some of these things do or don't have particular stable structures. So they actually only are going to have a particular structure under very specific conditions. And if they only have a specific structure under conditions, they're only going to have a specific function under specific conditions. So that's supposed to be shown here by the red squiggles. So red squiggles means doesn't normally have a particular structure unless it's interacting with a different protein, in which case it does form a structure. In some cases, you'll have modifications, and we'll talk about these covalent modifications again on Monday, which then those covalent modifications then give you a specific structure. Sometimes there'll be some kind of interaction which gives you a specific structure. And in a very few cases, just not having a specific structure does actually turn out to be important for function. And this is particularly important if you think about hydrophobic side chains, or particularly lots and lots of hydrophobic side chains. What are those going to do? Those are actually going to be interacting with each other, but not in a particularly defined way. And it turns out that not interacting with each other in a defined way is also really nice if you have to have something which has to be very dynamic, say a membrane. You know, in membranes, the hydrophobic interactions, membranes are moving around all the time. And if you need to have something inside a cell that you want to make sure has a lot of motility, maybe having some of these unstructured domains is important. It turns out that the nuclear pore complex is a really good example of this particular kind of unstructured proteins. So the last thing I wanted to talk about in terms of structured versus non-structured proteins, hopefully lots of you have heard about things like Alzheimer's and Parkinson's disease, etc. Some of you may know this has to do with amyloids. What's amyloid? Amyloid is when you have a protein that forms a particular structure which is then non-soluble. And it's actually, these are proteins that have multiple different kinds of structures that they can assume. It can either be one structure or a different structure. Mad cow disease is a really nice example of this, where you have one protein that can have two different structures, fine structure, structure which causes all kinds of problems. So that's the bad kinds of amyloids, because amyloids, they're forming these structures that all end up being filaments, precipitated proteins. Precipitated here is it's highly hydrophobic. That's going to interact with itself as opposed to interacting with anything else. More recently, people have found that actually some amyloids are actually absolutely important for function. And the best understood of those are these, which are proteins that have so-called low complexity domain. What the heck is a low complexity domain? What do we even mean by low complexity? So complexity here means amino acid sequence. So very small amounts of variation in amino acid sequence. But it turns out that these can actually, under particular conditions, form these things that look almost exactly like these amyloid plaques, precipitating, but in the process also pulling proteins and sometimes RNAs into particular places inside the cell. And this is something which is really hot research right now. People also call it phase separation. It's really thinking about oil in water. It turns out that inside cells, very often you'll have some oily regions, some non-oily regions. How do you get those oily and non-oily regions? It really seems to have a lot to do with this interactions of these amyloid proteins and particularly forming these hydrophobic areas. Clearly have to be very controlled, because they get out of control, you end up with things like Parkinson's, Alzheimer's, etc. But under controlled conditions, you absolutely require them. So with that, wish you a good weekend, and we'll see you on Monday.